Hello and Assalamu alaikum. In the previous session, we have talked about the structure of different types of lipids. We talked about the classification of lipids and have looked at their structures and also have talked about some of their functions as well. In this lecture, we are going to talk about lipids as constituents of biological membranes. This is again to briefly comment about the structure of membrane lipids. The different types of lipids which are present in the membrane. For example, some of them are glycerophospholipids. They contain glycerol, they contain fatty acids, and a polar head group which is attached through a phosphate. They're called as glycerophospholipids. In addition, there are sphingolipids as well. Sphingolipids contain sphingosine. Then there are glycolipids as well, which are conjugates of carbohydrates with lipids. So there are different types of membrane lipids which are present in the biological membranes. These not only include the cell membrane, but also include the membranes of the organelles. Nonetheless, whatever the type of lipids are present in the membrane, they are bipolar molecules. By saying the word bipolar, it means that they have two poles on them. One part of the molecule is hydrophilic, which is the polar head group. The other part of the molecule is hydrophobic, that is made up of the fatty acyl chains. The polar head group is hydrophilic and hence it is able to interact with water molecules around it. The nonpolar part, which is made up of the fatty acyl chains, is unable to interact with water. So therefore, these nonpolar parts of the lipids, since they are unable to interact with water molecules, so they are interacting with each other because there is solvent, which is water, all around them, and they're unable to interact with water. So because of this difference of polarity, they are unable to be solvated. Hence, the nonpolar parts of lipids are pushed to interact with each other. Therefore, the nonpolar parts of the lipid or the hydrophobic parts of the lipid, they assemble with other nonpolar or hydrophobic substances. This is a diagrammatic representation of a phospholipid molecule. Here, highlighted in red color is the hydrophilic head region of this phospholipid. And in blue color, you can see these two fatty acyl chains. You can note here that this fatty acid attached to one of the carbons is saturated fatty acid, and the one present here is unsaturated. So you can see the kink present in this molecule because of the presence of a double bond. So this part shown in red color is the polar head group. This is hydrophilic part of the molecule and hence is able to be solvated by water molecules. And the two fatty acyl regions, they constitute the hydrophilic tail, the hydrophobic tail of the molecule. This is a space-filling representation where you can see the fatty acyl chains represented here, and this part is showing the glycerol molecule. This portion is representing the phosphate, and to this phosphate, other polar groups are attached. In the previous lecture, we have talked about those different types of polar head groups which are attached to a phospholipid molecule. And this is another diagrammatic representation. Most of the time, when we show lipids in the membranes, we represent them like this. So this red circle here is showing the hydrophilic head region, and these two tail region, the so-called tail region, is representing the hydrophobic part of the molecule. So that's a cartoon representing the same thing, that the polar head group has an affinity for water molecules. But the hydrophobic tails here, they are unable to interact with water, so they repel water because they are nonpolar. This is a diagrammatic representation of interaction of lipids. So this is an assembly of lipids which form a monolayer. Since you can see that these lipids are arranged in form of one single layer, so they are called as a monolayer. Such monolayers of lipids exist when on one side, on one face of the molecule, there are polar substances. For example, on one side there is water available. The water molecules are polar, so the lipids are going to orient in this manner that their polar head groups are going to be projected towards water, because the polar head groups can be solvated by water molecules. The tail region is hydrophobic. It is unable to interact with water. It is being repelled away by the water molecules. So the hydrophobic tail region is going to assemble with one another in this manner that the tail regions are going to project towards air. This is a typical organization of lipids or other bipolar molecules at the interface of water, which is polar, and air, which is nonpolar. So at this interface of water and air, polar and nonpolar, the lipid molecules are going to be oriented in this manner that their polar head groups are going to project towards water, and their hydrophobic tail regions are going to be oriented towards air. So in this way, the lipids, they arrange themselves in form of a monolayer at this interface of air and water. In case of biological membranes, there is water present on both sides of the lipid assembly, of the lipid aggregate. Therefore, the lipids are going to be arranged in form of a bilayer. However, if one of the phase is hydrophilic and the other phase is hydrophobic, as shown here, water versus air, then lipids are going to be oriented in form of a monolayer. This type of arrangement of lipids is very important in the lungs. The cells of the lungs, they contain water in them. But in the alveoli, the regions where gaseous exchange is going to take place, so on one side of the alveolus is water, and on the other side is air. So the lipids present in the lung are going to be oriented in form of these monolayer-like structures. One of the very important type of phospholipid present in the lungs is called as dipalmitoyl phosphatidylcholine. As the name says that this contains a choline attached to the phosphate, which in turn is linked to glycerol on carbon number 3. Carbon number 1 and carbon number 2 of this glycerol, they contain two palmitic acid residues attached to it. So in this way, they both are saturated fatty acids. This lipid present in the lungs, in the alveoli of the lungs, it acts as a surfactant. It helps in decreasing the surface tension in the alveoli. Since these two fatty acyl chains, they are palmitic acid, they are saturated, so therefore they form an extended straight structure. They do not have any bending in them because they're saturated fatty acids. Now, this is typically important when the air is being expelled out of the lung. When air is expelled out of the lung, this would result in collapsing the alveoli, as air is going to be blown out of them. However, these saturated fatty acids which are present in this phospholipid in the alveoli of the lung resist compression, and hence it is easy for the air passageway to open easily. These saturated fatty acids, they assemble, they pack with each other in a tightly packed manner. Because of this, they prevent the alveoli from collapsing. Hence, when air flows back into the alveoli of the lungs, it is easier for the air to enter the alveoli and expand the lungs. However, when lipids are present in an environment where there is water present all around them, so they orient themselves in this following given manner. This is a diagrammatic representation showing one molecule of a phospholipid. So you can see that this is the polar head group, which is hydrophilic, and the non-polar tails, which are hydrophobic. Since there is water all around, so the phospholipids are going to assemble in such a manner that their polar head groups are going to be projected outward, facing water, and their hydrophobic tail regions are going to be interacting with each other. They are tightly packed with one another so that they exclude water molecules from the interior. In this way, these lipids are going to assemble in form of a bilayer. This is an electron micrograph showing the structure of the bilayer, and this is a diagrammatic representation here. The membrane is fluid in nature. Fluids are those substances which have greater degree of mobility of their molecules as compared to solids. So membranes are also fluid in nature. By saying the term fluid, we mean that the molecules which are present in the membrane, they have a certain degree of freedom to move around in the three-dimensional space of the membrane. The molecules of the membrane do not occupy a fixed position, but rather are dynamic. This means that these molecules they can move around in the membrane freely. Not only the lipids, but also proteins which are present in the membrane are able to move around. Many proteins form large complexes in the membrane, and they can move around in the membrane in form of that assembled complex. Some of the proteins and lipids, they form an aggregate as one single unit, and they all move around in the membrane as a single unit. These structures are called as lipid rafts. We will study about lipid rafts at the end of this session. So the membrane is fluid in nature, and the lipids and the proteins which are present in the membrane, they are able to move around in this three-dimensional space of the membrane. This is a diagrammatic representation which is showing the fluid nature of the membrane. Although it is not represented here, but the lipids molecules, the individual lipid molecules, they can switch places with one another. This is not shown in this representation. This is another representation showing the membrane. The lipid bilayer is represented here, and all these substances which you see around are proteins. So these are all membrane proteins. They're embedded in the membrane. Many of these proteins, they carry out many important functions. For example, some proteins, they bind to the molecules or other proteins present on the exterior, and as a result of which, they transfer that information to the interior of the cell. Some of the proteins which are embedded in the membrane, they are important to interact with the extracellular matrix. Some membrane proteins, they act as transporters. They form channels through which different types of molecules can move across the cell membrane. So this is the outside of the cell shown here, and this is the inside. These are the various different organelles and some other proteins which are present inside this cell. So the membrane is fluid in nature. In this representation, you can see the movement of a lipid molecule in a two-dimensional space. So this representation shows that the lipid molecule which is present here is going to switch place with the lipid molecule on its right, as represented here. So this movement of lipids within the same part of the membrane is called as lateral diffusion. The lipid layer is actually made up of two layers of lipids. The layer of lipids which is facing the outside is called as the outer leaflet of the membrane, and the one which is facing the inside is called as the inner leaflet of the membrane. So here you can see that diffusion is going to take place within the same leaflet of the membrane, so this is called as lateral diffusion. In this representation, it shows a 2D plane, but actually the lipid molecules or the protein molecules, they are going to migrate in a three-dimensional space of the membrane. The other type of movement is represented here, that is called as the transverse diffusion. In this type of diffusion or transfer, the lipids from one leaflet of the membrane are going to be flipped and they move to the other leaflet of the membrane. We will see about this later on, that the outer and the inner leaflet of the membrane, they contain different types of phospholipid molecules in them. And the way these molecules are going to be partitioning in these two leaflets is through this flipping movement, which is sometimes also called as the transverse movement or the transverse diffusion. So the membrane is fluid, and therefore the molecules which are present in the membrane, they can diffuse around in this three-dimensional space. The fluidity of the membrane depends upon many different factors. We have previously talked about the factors influencing the transition temperature of lipids. The same factors also apply to the aggregates of lipids in form of membranes. Different cells, they regulate their fluidity through several different mechanisms. We will briefly comment about that as well. One such mechanism is regulating the amount of cholesterol present in a membrane, because the presence of cholesterol decreases the membrane fluidity. So those membranes which have more cholesterol in them, therefore they are less fluid. Cholesterol is a planar ring structure and it is a rigid structure. So increase in the amount of cholesterol in a membrane means that this membrane is going to be less fluid. A very important technique to study membrane fluidity and dynamics, that is called as fluorescence recovery after photobleaching. Before talking about the details, let us first focus on the process of photobleaching. In order to study the dynamics of a given membrane, the molecules of the membrane, they're labeled with a fluorescent marker. This is represented in this figure. The fluorescent markers, they may be different types of fluorescence producing chemical substances. These chemical substances are available from commercial suppliers and could be purchased, and by using a suitable method, they can be added to the membranes. These fluorescent molecules could be used to label the lipids or to label the membrane proteins as well. Once the membrane molecules, they are become labeled with a fluorescent tag, so the membrane is going to produce fluorescence. This fluorescence is recorded by a camera which is associated with a microscope. So we look at the cell through the microscope and we find the fluorescence on that membrane. The term bleaching means to decolorize, to remove the color. When a substance in the membrane, say for example the membrane lipids, they are labeled with a fluorescent substance, so they're going to produce fluorescence. If we bleach a particular region of the membrane, then the fluorescence will be bleached or omitted from that particular region of the membrane. This is diagrammatically represented here. This is a cell, and the membrane of the cell is homogeneously labeled with a fluorescent molecule. The fluorescent marker is represented by green color here. So one of the ways to label the membrane is to put in different types of fluorescent chemical substances. Another way to label the membrane is by addition of fluorescent proteins. These fluorescent proteins are obtained from various different sources. Most commonly, they are obtained from jellyfish or some types of bacteria. So either by using proteins or by using different types of chemical fluorescent tags, the molecules of the membrane are fluorescently labeled. Say, for example, in this case, the lipids of the membrane are labeled with a fluorescent substance. So all of the surface of the cell is going to produce a fluorescence signal. The next step is to carry out bleaching. During bleaching, a laser is incident onto a certain part of the cell, which is represented by this white area here. So the laser is going to inactivate the fluorescent markers which are present on the membrane. Since those markers are going to be inactivated, so they no longer produce fluorescence. Hence, that is called as photobleaching. That is, the fluorescent markers, they're inactivated, so they are sort of decolorized. They are not going to produce the fluorescent signal anymore. And this is mediated by using a particular wavelength of a laser, so the process is called as photobleaching. In other words, it is sort of going to decolorize the fluorescent marker which is associated to the membrane. This is diagrammatically represented by this white circle, but in case of an observation through the microscope, this is going to appear as a dark region. Now, the lipid molecules which are present in this white circle, they are now bleached. That is, their fluorescent tag has been inactivated. Now, since the lipids are mobile, they're able to migrate in the membrane. So these lipid molecules, where bleaching has taken place, they are going to diffuse in the neighboring part of the membrane. And other labeled molecules which have an intact fluorescent label on them, they are going to diffuse in this white circle. So at a certain point of time when the laser is incident, the entire area will be bleached, and therefore the fluorescence intensity is going to be very, very low in this area. But as time is going to pass, the membrane lipids are mobile, so the inactivated lipids are going to migrate in the neighboring part of the membrane, and active label containing molecules from the surrounding are going to diffuse in this area. So therefore, because of the diffusion of inactivated molecules away and the labeled molecule towards this area of interest, the intensity of fluorescence is going to recover in this bleached area. So if the membrane is more fluid, that is the lipids, they can move around easily, then the time required for this diffusion is less. And hence, the recovery is going to take place rapidly. When I say the word recovery, I mean recovery of the fluorescent signal in the bleached area. However, if the molecules they're confined into a certain specific region of the membrane, or the membrane is not very fluid, it has a less fluidity, then the migration or the diffusion of these labeled molecules is also going to be slow. So now, after carrying out the process of bleaching, we look at the time which is required for the recovery of fluorescence in the bleached area. The time required for this recovery of fluorescence in the bleached area is going to provide us very useful information about the fluidity of the membrane. Greater is the fluidity of the membrane, that means more is the rate of migration, more is the rate of diffusion of molecules in the membrane. and hence greater is the rate of diffusion, so quicker is the recovery of fluorescence in that bleached area. And if a membrane is less fluid, so the time required for carrying out this recovery will also be greater. This is a diagrammatic representation. This is a cell which contains a fluorescently labeled molecule in it. So this part is to be bleached, so a laser is incident on the molecules present in this area. The laser is going to inactivate the fluorescent molecules in this region. So you see that once the area is bleached, the fluorescence intensity decreases a lot. But since the molecules, they are mobile, they're able to move around, so the inactivated molecules diffuse away from this region. And more labeled molecules from the surrounding are going to come and replace them. In this way, the fluorescence in this given region is going to recover. This is a graphical representation of the same phenomenon. On the x-axis is time, and on the y-axis is the intensity of fluorescence. So the cells, initially, they will have a certain level of fluorescence. Now, in a particular area of interest, we carry out bleaching by exposing that area to a particular laser, which results in inactivating the fluorescent molecules. As the fluorescent molecules are inactivated in that area, the intensity of fluorescence decreases. Now, as time passes, the inactivated molecules are going to diffuse away from the area of interest, and more fluorescently labeled molecules are going to migrate in the area of interest. So therefore, greater is the migration rate, rapid is the recovery of fluorescence in that area of interest. If a cell shows greater rate of recovery after photobleaching, that means the membrane of that cell is more fluid. Molecules in that membrane, they can migrate easily. And if the time required for the recovery is slow, that is more time is required for recovery, that means the rate of diffusion in the membrane of this cell is also low. So this cell has relatively less fluidity as compared to the other one. This can not only be used for looking at the rate of diffusion of the lipids, but this is also typically very useful in determining the ability of proteins and protein complexes to migrate within the membrane. Certain proteins, they behave individually in the membrane. Other proteins, they form complexes and they migrate with one another in form of a large complex. For example, proteins forming the flagella, ATP synthase, and many other such proteins, they form complexes in the membrane and they migrate as one single unit. So therefore, when we look at the rate of recovery after carrying out photobleaching, this provides us with quantitative data to study the migration and fluidity of the membrane. The fluidity of the membrane is affected by several different factors. One such factor is the presence of cholesterol. More cholesterol means less fluidity of the membrane. Another important factor which affects the fluidity is the temperature at which the cells are operating. As a general rule, when the temperature is less, the fluidity of the membrane is also less. This is diagrammatically shown here that above the transition temperature, the lipids have sufficient amount of energy and they're more mobile, they're moving around, so therefore their packaging is less tight. Below the transition temperature, the lipids are going to be arranged with one another in a more tightly packed fashion, and hence they form a gel-like solid. But above the transition temperature, they are more fluid. They have the ability to diffuse around rapidly. The factors which affect the transition temperature of the lipids, they also affect the fluidity of the membrane. The transition temperature increases with the chain length. That is, if a membrane contains short-chain fatty acids in it, its transition temperature is low. And if it contains long-chain fatty acids, then the transition temperature or the melting temperature is also high. It is also affected by the degree of saturation. If unsaturated fatty acids are present, then melting temperature is less. If more saturated fatty acids are present, then the transition temperature will also increase. By regulating these factors like the chain length and the type of the fatty acids which are present in the lipids, and by regulating the concentration of cholesterol in the membrane, cells can regulate the transition temperature of their lipid bilayers. Under various conditions, the composition of the membrane can change, and this change of the composition of the membrane would result in regulating its transition temperature as well. Okay, so this is a slide from the previous lecture shown again here, that membranes are important for various important functions. One of the significant thing which the membranes are doing, that they are acting as a scaffold for various processes to occur. One such process is the transmission of nerve impulse. Nerve impulse is transmitted in form of electrical current, and this electrical current is generated by a potential difference of ions across the membrane of the nerve cells. Anesthetics, for example, these are the substances which interfere the transmission of nerve impulse. Substances like ether, cyclopropane, and several other such related compounds, they are uncharged nonpolar substances, and they're able to diffuse through the membrane. They interact with the nonpolar part of the lipids, and therefore they alter the membrane structure. Thereby, they impair the, the transmission of nerve impulse. So these gaseous anesthetics, they exert their effect by altering the structure of the phospholipid membrane. We are going to continue this lecture, and in the coming part of this lecture, we are going to explore more about the membranes, and later we will also look at the other different roles of lipids as well.